Thank you for your kind introduction. Good afternoon, ladies and gentlemen. I'm going to present you some of our work with my colleagues at the Institute of Radio Protection and Nuclear Safety in France. And part of this work has been also performed under a number of European projects. I will introduce you very briefly. Okay, so I'm really too far to see the screen. I can move. Okay, I'm not linked to the, sorry for these logistics things. So that's pretty much better for my vision there. So let me introduce you some basic lines of methods to demonstrate the protection of the environment, and you will recognize that many of them are quite similar to human health impact assessment or risk assessment. Ecological risk is an estimation of the probability and magnitude of the adverse effects likely to occur in an ecosystem or part of these ecosystems, together with the identification of uncertainties. And for performing any ecological risk assessment, it is needed to link exposure to radioactive substances, to effects, exposure to those, and those two effects. The basic components of any risk assessment are shown on this simple figure. On this side, you are going to perform the exposure analysis. On the other side, the effects analysis based on the knowledge you've got on those or dose rates effects relationships that will be used in order to derive effects benchmarks, as was explained a little bit by David previously. And you are going to combine the two information into a risk, and the simplest way for doing that is to establish a risk quotient in between the exposure and your effects benchmark. You will understand very easily that the robustness of any ecological risk assessment is highly dependent on both those estimates and on wildlife effects benchmarks you are going to adopt. ICRP 108 recommends to perform the demonstration through the use of a set of reference animals and plants and reference organisms. But in any case, it's clear that extrapolation rules are needed for both exposure analysis and effects analysis, and I will focus my talk mainly on effects analysis. So what about effects benchmarks for wildlife, and where are we now? Under the ERICA project, which were a project funded by the European Commission and then later under the AEA MRAS 2 effects group, we have prepared a number of effects data and built those effects relationship in order to propose a methodology to derive effects benchmarks, such as the one that has been promoted by the European project ERICA, the predicted no effect dose rates, which has to be interpreted as a dose rates below this value of 10 microgray per hour, which is unlikely to have any population effects, and this is used in any ecological risk assessment to screen out all such exposure situations. On his side, UNSCARE has also published recommendations, such as, for instance, the 100 microgray per hour to the most exposed individuals that would have unlikely effects on most terrestrial communities, and based on literature review, actually there released a number of such recommendations for different wildlife groups. ICRP has proposed, as explained by David, DCRL for RAPs. These are bands of low probability of effects to be used as points of reference. All those effects benchmarks are based on analysis of various dose-response relationships for a variety of species, from plants, invertebrates, and vertebrates wildlife groups, and for a variety of biological endpoints covering mortality, morbidity, reproduction, and mutation. And several methods also have been used for this meta-analysis of dose-response relationship. For instance, per species representative of a family of species. This is done under the ICRP approach. Or per set of species representative of the biodiversity existing among a larger assemblage, among a wildlife group or an ecosystem. This is the approach that was selected under the UNSCARE recommendation and the ERICA Protect projects. But in any case, once again, they are all based on a number of extrapolations. So the various extrapolation and associated uncertainty are listed here. The response in terms of from biomolecules to ecosystems, in terms of structure and function, varies to radiation, varies in nature and in intensity according to a number of items. I'm not going to go through them one by one, but will, in the follow-up on my talk, give you illustration of the last three of them, among levels of biological organization, and I will mainly comment this shift from effects observed at the individual level to the population of a given species, which is the most important one, I guess, for this field, for radioprotection of the environment, among species, and from effects data acquired under laboratory control conditions versus the real world in field. So let's start with the first one, from how to propagate effects observed at the individual level to the population level. So the principle is that for a given species, it's possible to combine individual dose-response curves to infer the population level by using the Leslie matrices, which is a tool that allows you to simulate the demography of the population of a given species. To do that, you need to have a simplification of the representation of the life cycle of your population and to divide your population into several age classes, each of these classes being characterized by a number of features. So first of all, you have to divide the life cycle of the species of interest into 3 life stages defined as egg stage, juvenile stage, and adult stage, with events separating the two, such as hatching or birth and sexual maturity there. After that, it's possible to give features to the different age classes in your population, defining the survival rates per classes and the fecundity rates for adults. And once you have done that, actually you've got the parameters of this Leslie matrix that allow you to, from the knowledge of the number of individuals constituting a population at a given time, it's possible to calculate the number of the individual of the population at the given time plus one time step. Then it's also possible to combine the information of radiation effects to those features. For instance, here, dose rates effects relationships obtained on hatchings can be applied here to simulate the reduction in survival rates in egg stage. And you can follow the same reasoning for effects on survival when you are going to apply this as a reduction in survival rates in juveniles and adult stage. And finally, the effects on fecundity will be applied as a reduction on fecundity rates. So at the end of the day, you obtain this type of information. You are able to combine observed effects from ionizing radiation among several individual endpoints. You are able to model the population growth rates and the individual reproductive rate, which is the number of offspring per individual over a lifetime at any dose rate. And you are able to predict dose rates causing population extinction, which is a population-relevant endpoint that is of importance to interpret the ways the population of species evolves under any stressor in the environment. So this will allow you, for a given species, to compare between the radiosensitivity at the individual level and the radiosensitivity at the population level. Actually, this is not so simple because population level effects depend both on how key biological function are affected by radiotoxicity at the organism level, and how population dynamics, different age classes, will respond to such effects. Indeed, the same level of effects could have different consequences for the population, depending on the impaired individual endpoint and on the life history strategy. The most influential individual endpoint for population dynamic is not necessarily the most radiosensitivity. And we performed a modeling exercise on various species under a European project to demonstrate that there are actually three types of situation. One where the most radiosensitive individual endpoint is an overestimation of the population radiosensitivity, or a good proxy of the population radiosensitivity, or an underestimation of the population radiosensitivity. So we do not have only one answer to this question of the propagation of effects from individual to population level, but several. Let me give you an example to illustrate that. The first example deals with a terrestrial mammal, the mouse, which life cycle strategy is very briefly written down here. On the left hand, you've got dose rate effects relationship on individual endpoints with the effects on fecundity on the mouse, on hatching, and on survival. And just to help you in having a quantification of what I'm telling about, you've got here the value of the dose rate giving 10% effects, for instance, here on the litter size, the effects on fecundity. Then, according to what I have explained previously, you're going to combine those effects into the Leslie matrix to obtain the consequences at the demographic level of the population, and you will obtain those values. And you will see that the dose rates giving the lowest significant change on the reproductive outcome at the level of the population and of the growth rate of the population are, in that case, higher than the most radiosensitive individual endpoints by a factor around 10. So that means that in that case, the population of mouse is protected when individuals are protected. And if we have a look on the ICRP DCRL reference rat, which is of the same family, you will find that the proposed bands is appropriate for the individual level, and the upper bound will be appropriate for the population level. So this bands is appropriate for individual, but overprotective of the population level. The other example, exactly the same system for reading that. It's for terrestrial invertebrates, the earthworm, which is one of the RAP. So the individual endpoints on your left hand and their dose rate. And here the combined consequences for individual reproduction rate and the population growth rates. In that case, the dose rate giving the lowest significant change in R0 or in lambda in the population growth rate are lower than the lowest individual value of EDR10 at the individual level. So the population is not protected when individuals are protected. But the ICRP DCRL for reference earthworms gives a bands varying from 10 to 100 milligrams per day, and actually it is appropriate for individual and population levels, but we've got this information in addition. Now let me comment very briefly the interspecies extrapolation and lab-to-field extrapolation issues. Laboratory data have been preferably used since laboratory conditions offer a robust way to characterize cause-effects relationship. This was adopted from the EC-funded ERICA project suits to derive screening benchmarks consistently, actually, and this is important with the approach applied for chemicals, where laboratory data tests have been the main basis of benchmark until now. However, it is clear that laboratory and semi-field tests constitute a very simplistic way to represent the complex nature especially because they ignore inter-individual and interspecies interactions. They ignore the variety of routes of exposure and the variety of responses from all species. So field data, on the other hand, are obviously representative of real work, but they always document ongoing and past changes, and the observed effects there may be caused or modified by simultaneously occurring stressors, and like in any eco-epidemiological study, you've got the issue of confounding factors, which is a really big issue. So here it's a representation of the radiosensitive variation between species and the chronic gamma controlled exposure in the lab. So each symbol represents here one species and the lowest value of the 10% dose rate, giving an effect on population relevant endpoints. And all species have been allocated to a wildlife group among plants, invertebrates, and vertebrates. So you see that the radiosensitivity of the species vary all over 6 orders of magnitude in between the most radiosensitive to the most radioresistant. If we do the same meta-analysis for data acquired on field, and especially for data acquired under the Chernobyl exclusion zone, the last 15 years, we have observed a shift of the sensitivity on the left tail by a factor of 5 to 10, and probably meaning that maybe the species and the real world should be more radiosensitive, but also we could not screen out bias such as bad dose rate or dose estimations, or bad sampling strategies such as avoiding the seasonal cycle effects or other factors such as other toxicants. So finally, to conclude and give some perspective, so it's clear that predicting radiological consequences at the population level, which is the one of major interest for radioprotection on the environment in wildlife species, requires knowledge on exposure conditions, on life history characteristics of the organism exposed, integration over time of a time-dependent absorbed dose, and combination of dose-response relationship on impaired individual endpoints. What we are going now to study, we are starting to study, is also the multi-generational effects, because effects propagating through multi-generation, through a real chronic exposure, could change the sensitivity of the species to ionizing radiation. So we try to promote at RSN at the European level research on primary mechanisms of interaction between ionizing radiation and living organisms, from biomolecule at least up to population. And for doing that, we are trying to use system biology-like approach such as dynamic energy budget model, which describes how individuals acquire and use energy based on simple rules of metabolisms, and how any toxicants or any stressors like ionizing radiation may alter the energy acquisition and the allocation of energy to growth and reproduction. And on this basis, it's also possible to have the consequences at the population level. I think that's it. I thank you for your attention, and I take this opportunity to share with you this web link, which is a link for radioecology exchange, where we put a number of lots of information and news that comes from the European Radioecology Alliance, which is the platform of radioecology in Europe that has been established legally last year, and research news from the Network of Excellence, STAR, and the Newborn Comet, which is a recent project that is going to implement joint programming mechanisms at the level of Europe at least, and in the field of radioprotection. I really thank you for your attention.